Even if it feels like you can make that.
Stay on time. I have the great pleasure of introducing Henry Adams, our seminar speaker today. He did his master's in forestry at Northern Arizona University, followed by his PhD in ecology and evolutionary biology at the University of Arizona.
Yep.
He is a forest ecologist. Comes to us from Washington State University, where he is an associate professor in the School of the Environment. He's been there since 2020, but he's been a faculty member since 2015. So Henry's work really focuses on tree mortality. He's paved our way for understanding tree mortality, and much of his work has looked at physiological mechanisms underlying death from things like drought, heat, fire, pests, and pathogens. He has a really impressive breadth of work from things like stress physiology, but also phenology, scaling trees to forest ecosystems. And I've personally admired his work since I was a grad student and continue to be inspired by him, and hope that the lab is also inspired by his work for the things that we're currently thinking about. What else I want to say? Oh, Henry gave me a menu item of things he could talk about. And one topic that I chose, I'm really excited about, is fire-induced mortality. And I chose it not knowing that we are going to be the first audience to hear about this work from Henry. So I think that's really special, and I hope you're as excited as I am. And I wanted to just make a plug for one thing. Tomorrow we are going to have a journal club with Henry in Lilly 1136 at noon. So you're welcome to join. If you would like the paper that we're going to be discussing, send me an email. My name is Morgan Furs. You can stop me out of the seminar to get that as well. But also come, even if you haven't read the paper, you can ask Henry questions from the seminar or just about ecophysiology in general. So please join me in welcoming Henry.
Thank you. That was really, really kind. I don't know if I paved any roads, but I followed them. They took me cool places. Yeah, so trees, drought, beetles, fire, and death. Sorry, you could get right to the point, but I got your attention, didn't it? And Morgan, you have a great introduction of like my past, but I just kind of threw this in. I had this for another slide, kind of another presentation, but it's kind of cool. Like, you know, you might be wondering why this guy from Washington State University keeps talking about New Mexico. Now you know why. I did the majority of my training in the Southwest US, including postdoc at Los Alamos National Lab. Then I was 5 years an assistant professor at Oklahoma State University before I moved up to Washington, and I was regularly making this road trip to continue work in New Mexico. Cool. I'm from upstate New York originally, and went to school up there. So that's about me. But back to the whole point: drought-induced tree mortality, and this is a hill slope in northern New Mexico. 2002 was the height of a pretty severe drought. Actually, I was at NAU at Flagstaff in northern Arizona at this time doing my master's degree, and it was the hottest, excuse me, let me rephrase that, it was the driest year in Flagstaff, estimated from tree rings, in 1,500 years. So it's pretty intense. And this is what I would later go to do a postdoc, like just over this hill here, but that was 10 years later. This was happening. I saw this sort of thing seen play out in Flagstaff too. There's nothing wrong with the color of this photo. Which is really funny that I'm telling you that because I'm red-green colorblind. But it does look kind of brown, right? That's what I'm saying. You're seeing that too. Great. So these trees are all dying from this drought, which happened during a period of relatively high temperatures. There was also this guy that showed up, Ips confusus, the pinyon bark beetle, that was contributing to tree death. And that looks kind of bad, but it just sort of looks like an old, wrongly colored photo. But what if you went back to the same place 2 years later, and this is what you would see? Those needles fall off, you get this really vastly transformed landscape, right? I would imagine any property of this ecosystem that you might be interested in, there's a good chance it's been affected by this die-off, right? We're not talking about background tree mortality, right? Trees are born and they die all the time, right? Some die, some live, that's life. This is a whole hillside, right? Most of this Pinus edulis, pinyon pine, Colorado two-needle pine. Following a little later on are some ponderosa pines that were grown at this site. The green left here is Juniperus monosperma, much more drought-tolerant species that can occupy lower elevations and is persisting, but in some of these sites, actually, a 2011 drought came for some of those as well. So this is kind of like interesting happened in the backyard of some scientists who happen to already be thinking about these questions like, what does it take to kill trees from drought? Is this something we're gonna be seeing a lot more of with climate change and what's going on? So it led to some cool ideas. But when this was happening, it wasn't just one, here in Indiana, you're like, okay, cool, it's just one random hillside in New Mexico, so what, right? But it was pretty widespread. So here's a map of Arizona, New Mexico, strip of the states to the north there, and in green, all the forest and woodland. You go, really? I thought it was just like all desert there. No, there's actually considerable amount of high elevation forest and, sparsely wooded woodlands. And here's a cool point. This is all the forest mortality in both the forest and the woodland over in a 12-year period, 97 to 2008. It affected 11% forest and woodland. So here's where that hill slope I just showed you, that was here, pretty well hard hit area. Not every place here was a 90% mortality of one codominant species, which was the case at that site. But some places were pretty heavily impacted, as I just showed you. But it's bigger than this. It's not just across the Southwest US. This was part of a larger conifer die-off across the Western US. So this is by my colleague Arjen Meddens at WSU, who made this a while back. And this is categorized by different species of bark beetle. You can see mountain pine beetle showing up. There's the pinyon ips in blue down here that was causing the mortality I just showed you. And it's bigger than just Western North America. Recent study that I'm not going to dive into, a recent few years ago, by my former PhD student, Bill Hammond, was putting these cases together from around the globe over the last 48 years or so, 48-year period or so, and showing that this is quite common worldwide and that it's linked to hotter temperatures, drought, and attack by biotic agents like those bark beetles, sometimes fungal pathogens. So it's this interesting phenomenon that's going on, and there's a lot of questions about, well, is this going to get worse in the future? Like, what's going to happen with climate change? We expect it to get a lot hotter. We expect that droughts could increase. We'll have big swings between hydrological extremes of drought, fluvial periods with lots of moisture. And so there's some interest in trying, like, what can we do to kind of like understand what's driving this and how we might better predict it? One of the ideas for this that I'm going to go into today, I have a whole other seminar given on like more empirical methods, just correlating climate conditions with where we see trees dying. But physiological mechanism is kind of one of the like sexiest ways of like, what if we like knew, like, right? Everybody loves physiological mechanism until you really start diving into it, because the problem is that everybody's mechanism has another mechanism. Pretty soon we'll have subatomic particle physicists, but a little bit of mechanism might go a long way. In fact, we'd be better do that, right? I got good advice once from Steve Running, ecosystem modeler. I think he's retired now, who had said, you need to go like 3 levels of mechanism below what you use to predict. So even if we do end up using empirical predictions are the best ways to do this, we should know if they're any good or not, right? Otherwise, we have like the correlation, not causation, and extrapolation. Those are not good things. So if we want to have physiologically predictive models of where trees die, then we need to understand something about the physiology of how trees die. And this idea came out in 2008 by a guy named Nate McDowell, was my postdoc advisor. Some call this the paper that launched 1,000 studies. I heard that term. If you haven't seen this before, this thing's cited like 8,000 times now. What irks me is people cite it as it's the final word on how trees die from drought. I'm like, we're just getting started, I'm always the reviewer. You might want to, if you get that comment on your paper from me, you might want to go look at like what we've done in the last 14 years. You can't just use this to answer your question. This was the hypothesis being proposed. We had these 2 ideas, and you can see that they depend maybe on the duration of water stress and the intensity of water stress, right? And you have these 2 endpoints, carbon starvation, and this one in green, hydraulic and some plastic failure. I'll just call that hydraulic failure. We acknowledge that when the xylem fails, that's going to lead to the leaves drying out. And both could be amplified by biotic agents that's applied here. So what are these things? So I want to tell you, like, physiologically what happens in a tree while it dies. You might want to think about physiologically what happens in a tree when it's not dying. Let's start there. So hydraulic failure, what's that all about? That's about how the xylem works, right? Trees supply water to their canopy by pulling it into the roots from the soil, right? Up the roots, up the stems, out into the leaves. And that water is actually being moved out into the atmosphere, and this pull is generated by atmospheric demand for moisture. Right? As long as we're less than 100% humidity and the sun's shining, even a little bit of intensity, that'll drive that, right? We're not too cold. this is going to happen. So this whole system is under tension. This water is being pulled, a continuous column of water being pulled up tree. Now, what happens? Well, this is the everyday situation, right? It's working great. Trees get to move their water up for free. Very little energy goes into this. What happens if we throw a drought on top of the everyday situation? Of course, throw a drought in, we are going to do two things. One, increase the atmospheric demand for water, and two, decrease the soil moisture supply. So we're going to pull harder on that continuous connection, that rope, if you will, of water molecules. If you pull hard enough on something, put enough tension on something, and that's exactly what this is, you can break it. In this case, how does it break? Air embolism, or you might call it cavitation. Some people like to argue about the difference between cavitation and embolism. Maybe some in this crowd know about that, but I'll just use the term embolism because everyone tends to agree on the difference there. So you get enough air blockage of the movement of water over enough of a cross-sectional area of a tree, that leads to death. That's what hydraulic failure is. But as I like to tell my students, trees are not stupid, right? They're going to do everything in their power to prevent this from happening. As Anju knows, right, they have these things called stomata, and most of you have heard of them before, right? Why not just close the stomata? That ought to be a good way to avoid these tensions. That might have some other consequences, right? First, I wanted to show you a picture of what this looks like. I forgot that was there. So we can take a red safranin dye, or a safranin dye it is red, and when you run it through, let it perfuse through a stem, then take a cross section, you'll see it react with all the lignin in the xylem conduits, and the tracheids in this case, in this loblolly pine. So when you have a plant that has very little embolism, very little blockage of flow, it'll look like this. And if you have a lot, it'll look like that. See all the secondary xylem here, it's green, should be red, but the dye could not pass through. So you can kind of visualize it looking like this. Enough of this should be lethal at some point. So as I said, trees might have some ideas for how to avoid that problem. And that's where carbon starvation could come in, right? So everyday situation, you have photosynthesis, bringing in carbon, fixing carbon, that goes into non-structural carbohydrates as a storage, that's your sugars and your starches. And then trees use that, right, to pay the bill of respiration as building blocks for carbon skeletons to make everything the plant is made out of, right? However, close the stomata to avoid hydraulic failure and avoid those high tensions, no photosynthesis, because the water goes out through the stomata and the carbon comes in through there, right? Now, they have to rely on those stored starches and sugars in order to meet the demands of respiration. The idea with carbon starvation is once that gets below some critical point, they will die from carbon starvation. And I'll point out there's this space where perhaps both is happening in this original conceptual diagram. So yeah, I can simplify it even more. I tend to break it down into we have carbon starvation, hydraulic failure, and biotic attack, all probably potentially, these are all hypotheses, right? Interacting with each other and leading to mortality. So it's kind of a complex situation. I did some early experiments trying to poke at the answers to what was going on here with some transplanted trees brought into Biosphere 2. It's fun because these are like, what, 2 meters tall, but they were reproductively mature and 20 years old on average. They're a very slow growing tree, that 2-needle Indian pine. Did this inside Biosphere 2, actually, as part of my PhD work. We put the trees in areas of the glass house with 2 different temperatures. One was pretty close to ambient growing conditions for where the trees came from in northern New Mexico. And the other was where we cranked up the heat by about 4 degrees Celsius, start the experiment, and we stopped providing water to half the trees in each area until all of those trees died. And here's what we saw. This is a measurement of stem water potential. It's essentially the inverse of that tension, or if it helps, as you go down the y-axis here, that tension is increasing, right? And this is over time. And you can see they tracked fairly well together, the 2 temperature drought treatments, as they declined. On average, warmer drought trees died in week 18, and ambient drought trees, I think it's week 27 or so. And there really wasn't much, we don't have any actual significant difference here. So they were kind of doing the same things. I'm not going to talk too much about the temperature differences there, but it was interesting that there was a survival difference. But they both had high tensions, that really low water potential, that should be enough to cause complete embolism in these trees as they died. We also measured non-structural carbohydrates in the leaf in this experiment, is what I'm showing you here. So this is initially what we saw in terms of sugar and starch concentrations when the trees, just before the trees died from drought. You want to catch that just as they're going, which we defined as 95% foliar browning, if you're interested in that. We had a reduction that was significant. I think more important too is to compare to what the water control trees were doing at the same time. Take into any phenological changes that could have been happening. You know, photoperiod is a strong influence of this. Biosphere 2 glass house, and we don't have perfect control of temperature in a structure like that. So we had actually a 50% reduction in sugars and starches when trees died from drought. So we kind of have some indications that maybe both are going on, which was really cool. This was an early experiment, early data to come out on this, but I wasn't the only person doing this kind of data. So, and of course, we have, you know, trees dying around the world. From part of my postdoc, I got to do this global synthesis of tree mortality mechanism by pulling together data that other people had collected in similar experiments. So we actually ended up in total pooling the data from 19 studies on 26 different tree species. We got conifers and angiosperms in here, trees from various life stages. I'm not going to go into all the details of the study, but that publication's out there if you want to check it out. Focused in this work on what's the percent loss of conductivity at or just before mortality. PLC is measuring not the embolism, not like I showed you in those images, like what's green versus red, but more of what's the impact on actually the volumetric flow rate through the xylem conduit, right? Because you can imagine different size pores, some might embolize earlier than others, and that's going to have differential effects. So we don't do area, you verify with those area measurements, but we actually get in and measure the actual conductivity movement of water, and then estimate the maximum, and from that determine this measure called percent loss of hydraulic conductivity. And we also focus on non-structural carbohydrates, so sugars and starches that might be important in carbon starvation, right? I won't show you all the data. You'd be surprised how many of these studies early on, people got kind of excited about carbon starvation, which is really actually quite hard to measure. Non-structural carbon. So many labs spend a lot of time doing that, and they would do that and either not measure PLC or not measure or not have a vulnerability curve, which you could use to say convert. You can estimate PLC from water potential if you have something called a vulnerability curve. You can ask me about that if you want. So I'll show you in a moment. We had 13 cases that had data for both PLC and C. We'll compare. Going back to the hypothesis, you know, we had these two axes. We had the carbon starvation over here, hydraulic failure down here. I'm going to change up the axes and with the data that I had, and we have percent loss of conductivity. It's going up as you go this way, right? This is increasing embolism. So going from a state like this to here, right, to up to 100%. And if we go this way, on the x-axis, I have this non-structural carbohydrate deviation from control. You can imagine different plants, different tree species, they might have and need different levels of sugars and starches in them, right? So what we did is not compare raw data. We normalized what was happening in the trees that died just before they died to what was happening in the control treatments. So some experiments had water control, some had ambient trees in the field that did not die, so survivors. And whatever we had as our best control treatment among all those studies we synthesized, we called zero basically what the controls were doing, and then you could go this direction. Let's say we're up to 20% in a positive direction. That would mean the trees that died from drought had higher sugars and starches. If we go in this direction, we're saying the trees that died had lower. Thus, this is sort of our carbon starvation axis, right? Over here, we think carbon starvation. And of course, we could have trees die from hydraulic failure or carbon starvation like this, or from both. We could go in that direction, right? From a state of being alive. And here's the data where I found it. First thing I'll point out, I don't see any data over here. That's the point here. Hydraulic failure was universal. Trees were dying with relatively high PLC in this data set above 60%, 60% or higher, but some had increased non-structural carbohydrates, some had less. So that's pretty cool. Actually, if you're going to make models, predictive models of this mechanism, I would go with hydraulic failure, not carbon starvation, as the way to model that. And we're currently actually, I had a PhD student from China who's been visiting my lab. He just made the trip back. He's still working on this project. We had this preliminary data. We're actually expanding analysis because people doing these experiments kept going. And so all the open circles are from the original study I just showed you. The closed ones, Renyo put together. So we're seeing the same pattern. We're getting a little bit more spread across the data. And we're going to take a little closer look at these and say, did you really nail the right, like, is that I can measure before death. I can do that months beforehand and get this is relatively low PLC number. So I'm a little suspicious about these, but we're not seeing basically anything over in this realm over here. So hydraulic failure, definitely the way to go. And what I wanted to say about this too, is that, people used to say, I used to be accused in the literature that this doesn't happen under drought. My critics can no longer say that. I have demonstrated that. Now they say it doesn't matter. And they could be right. Stick with hydraulic failure. That's my point here. They could be right. I don't dispute that. What am I missing? Oh yeah, these guys. So how about some biotic attack? That might change the story, right? You can imagine it's kind of hard to put together, certainly if you're trying to do greenhouse studies. So that wasn't going to cut it entirely to study this. And I'll point out, as I said earlier, biotic agents is in here, is there some kind of amplifying factor, right? And there it is. Of course, the McDonald 2008 hypothesis has been revisited in more recent years. We can, if you think my work's very simple, we can make it more complicated if you'd like to. And we can think about these coupled water and carbon pools and how they function. We wrote this whole review on this. Is really interesting. And then where did biotic agents end up? Oh yeah, now they get a question mark. That's the progress since 2008. Cool. All right, so maybe that's somewhere we should look. Like, is that going to change the game? So to test that, we were lucky to get an NSF project, and we set up a field experiment at the Sevieta Wildlife Refuge, National Wildlife Refuge in New Mexico in a pinyon juniper woodland. We had a number of trees. This is our control. Nice, beautiful PJ woodland. We're just measuring the trees, not manipulating it in any way. And then we had these rainout structures that had these troughs over our plots that would capture a percentage of precipitation. Capture the rain in the summer. We even get snow here in the winter. There is some. One treatment was at 45%. Actually, we had two of these. It was really cool. We had a legacy treatment that had these structures on for 10 years. That's actually pictured here before we installed the other ones, including this new 45% treatment. Then we had a 70% approximately reduction of precipitation. Looks like this. And a 90+%. We really wanted to nail these trees. I've been on an experiment that only had the 45%. That doesn't always cut it. Sometimes it rains in New Mexico. So we wanted to make sure we saw a wide variety of tree responses. And in fact, on this one, the only place where water can get through is right around the canopies of the trees where it can get in. Only when it falls directly. We have these, hard to see, but you have these tubes that will pipe the water that's coming down the trough around that gap. And these work pretty well. I'll show you in a minute why, how. What do we measure in this experiment? All that primary physiology stuff that I said was important earlier. I'll show you data on water potential and non-structural carbohydrates today. But we threw in secondary metabolism. Let's get at the defense against bark beetles and the fungi they carry. So we measured monoterpenes, phenolics, and I'll show you data on resin flow. We also measured sesquiterpenes. I'll mention it, but I'm not going to show all that data today. Cool. This was a collaborative project. Not everybody pictured here, but some of the main players. Dr. Amy Trowbridge at University of Wisconsin was PI on this project, chemical ecologist, a good friend of mine. My former PhD student, Dr. Alex Thompson, Amy's former PhD student, Dr. Shayla Malone, and our former project postdoc, Dr. Drew Peltier, now assistant professor at University of Nevada, Las Vegas. So kind of cool. Everybody got a career advancement out of this. And I got tenures. There you go. Yeah, so all of these are a group effort. You need to show those pictures, right? So this was awesome. Have you ever gone into your experiment and gone, oh no, all our plants are dead? I go, oh yes, many plants are dead. Yes, we lost 40% of the study trees in 2021 and 2022, and we were stoked. Because as I said, you can do these experiments and nature's not with you, and it's a wet set of 3 years. That had happened to me during my postdoc. So this doesn't, I don't know, are you convinced that these are dying back here? You're not red green colorblind, you maybe are. I don't know. I put this is a nice picture, but here's a close-up on some trees. Sorry, it's coming out kind of blurry in the 90% plus. Yeah, we killed 40% of them. Ips confusus was in the traps that we put out there. We verified it as we saw it on the trees. It's so tiny, it's really hard to see the entry holes. Exit holes we were able to catch. And this is the number of dead trees and which treatment they occurred in. It was interesting. So we didn't lose anyone in the control. Well, we did kind of early on, but we replaced that control tree. None of the trees of the legacy 45, these trees that had, by 2022, they'd had 13 years of drought. and none of them died. How cool is that? The new 45%, we lost nearly all. And we've got half in the 70%. And some actually, this is 2021. It's kind of covered up by that thing there. We lost some in 2021 and 22 in that treatment. And so you gotta be careful too, if you set up experiments like this, like don't expect them to behave like perfect little experimental units. We don't expect that in the greenhouse, right? So we set these treatments up to create variability. So I'm not going to show you comparisons across treatments because different things happen. What's more interesting, what happened to those that died versus those that lived? And so here's what we saw in terms of pre-dawn plant water potential, right? Remember, as you go down this way, the tension is increasing and hydraulic failure is becoming more likely, embolism building up, right? And I have 4 lines on each panel here. Control trees, legacy drought, which did not die, then all the other drought treatments pooled together, those which survived versus those which died. And going all the way back to our pre-drought treatment installation in 2019, at least for the new plots, of course, the legacy would have had the structures on here. This is when we install in about January 2020, just before the pandemic, which was kind of cool. That might explain this gap right here. What do you think? Before I could get permission to go camping out here by a tent on my own, I mean, we were all like, we touched the gas pump, like when we drive out there, right? Those are some interesting times. So what do we see? Non-structural carbohydrates, those trees which died from drought, we actually got differences, significant differences. What, up to 2 years? Or let me rephrase that, up to like 15 months, bad, about 15 months before we saw most of the mortality or the last of the mortality. So they were low for a while. It was going up and down with the regular seasonal dynamics we saw in the other treatments, but they were significantly lower. And then they seem to have this crash in 2022. And then that's the end of the mortality we saw at that site. Cool. So we saw reduced, what did I say? Non-structural carbohydrates? What am I talking about? I guess that early flight tomorrow or yesterday was catching up with me. We saw hydraulic failure, right? It's very indicative of hydraulic failure. That correlates to a percent loss conductivity of 64%. Not surprising to us what we saw. So looks like hydraulic failure was definitely happening here. Okay, how about non-structural carbohydrates? Sorry if I misspoke earlier. How about needle sugars? We got a lot of interesting dynamics here, but no significant differences. Nothing to show you there. How about needle starch? Ooh, that looks cool. What's going on here? I'm not exactly sure. A good summer, perhaps. Good late, that's probably late monsoons were good. So the monsoons were late in 2021. That's the summer rainfall that comes to that region. So it had probably a lot of photosynthesis going on, but kind of cool, but no significant differences. All right, nothing happening with the non-structural carbohydrates. How about, or so far, how about twig starch? Nope, no significant differences. Are you getting bored yet? Needle total monoterpenes. All right, like the bark beetles were finishing off those trees. We're gonna start to see something, right? Oh, can you see why maybe this paper is now in review for the 4th time? This is not what anyone was expecting. These trees died from bark beetles. Wait, okay, maybe it's not in the monoterpenes, but I will say we looked at sesquiterpenes. In fact, it's almost 50 individual compounds, and there are like 3 of them that we found significant differences. And in those 3, the trees that did not die had lower in the non-exposed compounds. So that's how low. Nothing had higher. They lived. Not any of the 50 or so compounds. Amazing. How about phenolics? Let's look somewhere else. Nope, that didn't explain it either. Whoa, what is going on? It's kind of head scratching, right? Because bark beetles finish these guys off. So we look up the data a slightly different way here. This is twig monoterpenes, but not the absolute value, but as a percent of the control. So you have to imagine the control doing is sort of along this line, right? And here's the dying trees. And they had this interesting slope. So at really low water potentials near the end, they were cranking out defensive compounds like, oh my gosh, get it out there. But early on, they were on the low side. So there maybe is this idea they were it's a little too little and a little too late compared to the other trees and other treatments. How about resin flow, right? We're talking about the volume or mass of this sticky stuff that comes out, right? We measured that as well. The simple flow punch. Controls are here. Survivors, not significantly different. Trees that died. Drew is a real pro in statistical analysis. I'm not sure how he did that. I don't believe it. I don't buy it. First of all, legacy is even lower. Why didn't these die? None of these died, right? So I don't like these. I should just take the letters off of here. You can do all kinds of cool stuff with statistics, can't you? This guy's, this is Bayesian stuff. It's amazing. Yeah. But I'm not buying it. That's not explaining why they died. From another experiment in a greenhouse that we did as a companion study, we can find some insight that we think this was what was going on. I think this might be important. So what I'm showing you here is the induced response to both mono and sesquiterpenes pulled together in the same species, Pinus edulis, in a greenhouse experiment done at the University of Wisconsin. Yeah, they brought in pinyon pine by semi-truck. Pretty cool for that. After, this is the response, the change in these defense compounds in response to inoculation with Ophiostoma species. And you're probably drawn to the green and red dots. We had girdled trees. They also had their access to non-structural carbohydrates from the canopy cut off down to the site where the inoculation happened. That had no significant difference, by the way, on induced response, no effect whatsoever. So pooling them together, we do see an interesting relationship between the change in defense chemicals or the amount of inducible mono and sesquiterpene response and water potential. So as water potential declines, we get this decline in inducible response. And this is the approximate Turner loss point in pinyon pine and Pinus edulis. We look over here, we've actually estimated Turner measurement from osmolality measurements of the inner bark, phloem sap, and stem water potential. So it's estimating. It should be a positive number. It should not be negative. We know it's offset, but we are assuming it's offset evenly. So we also get a crash, right, in turgor pressure, estimated turgor pressure, as the water potential associated with turgor pressure, as that changes, crash in the defense. So this is kind of interesting. So maybe it's not like the amount of carbon you have to build these compounds, but actually the ability to deliver them to the site of attack, right, with water. So we're thinking that the same thing happened in the field. $1.4 million was not enough. We should have spent more. We should actually go back and do it again. Cool. That's what I wanted to say about drought. I want to shift it up. What am I doing on time? I'm doing really good. I want to talk about disturbance from wildfire. This is this new thing that I'm getting into, and this is going to be on the physiological mechanism of how trees die from fire. And sometimes when I tell my ecophysiologist colleagues that I'm studying that now, they go like, what? Why would you study that? Clearly, they just burn up, like, poof, into the atmosphere, right? Not a lot going on physiologically there. However, like these pictures that you find on Google, like this one I got from The Guardian and the Associated Press, I know this actually, that's outside Los Alamos. That's the Las Conchas fire in 2011. This is in Italy. If you're trying to get a story published, right, you're going to show the most extreme effect. If you're trying to publish a paper in a good journal, what else are you going to show on wildfire research, right? So if you read wildfire papers, you see pictures like this all the time because, look, this is important. It has big effects. But would you believe me if I told you that the majority of trees that die from wildfire in North America don't go out in such high severity impacts, right, with such high fire intensities? Check this out. I was driving out to a field site for another project in Northeast Washington, saw this, just like the sun was going down. We were trying to get to this site, check it out, and set up our tent before the sun went all the way down. So I didn't have a ton of time to explore. I saw that. I said, whoa, stop. Is this a whole bunch of like bark beetle drought-induced tree mortality that I didn't know about in this area? I got to go check it out. Got out of the truck, took a look around. I was like, huh, here's another picture, a little blurry. I zoomed in. What's all this black stuff right here? Those are all marks where that's a fire scar where the fire burned through. This is a wildfire. This is more typical, right? Sometimes this is called delayed mortality. But you have fire burned through on the ground, right? Kills the tree, but you don't find out until later. Canopy turns brown, falls off. There's some needles on here. See the gray stage in Douglas fir right here. Later gray stage, that's called. The needles were not scorched. They did not burn up the top of that tree. So it wasn't consumed in the fire. What killed these? And if you've ever seen this happen, there'll be a delay. Like it can happen. I saw it in Oklahoma in response to a prescribed fire to try to remove eastern red cedar, and it worked. It just took like 18 months. Then they all turned brown. You wouldn't have even seen signs of fire on the ground. So I have a good picture of that. I'm going to show it. So I've been working with a group. PIs involved are Alistair Smith, University of Idaho, Aaron Sparks, Dan Johnson. They set up these really cool experiments. These are a lot of fun. And we haven't yet been funded to study this mechanistically in large trees, but we're doing it in saplings. And we have these controlled experiments we can set up. this is really cool. So we have this combustion laboratory. No one's in here. This is a picture taken through a glass window from the next room over, where we set out, we put a sapling in a pot right here. This is lodgepole pine. Set up a fuel bed, and based on the mass of dried to less than 1% fuel moisture needles, and we're using specifically like hand-picked and carefully selected Western white pine needles, we know the fire radiative energy, the actual heat energy dose that this tree will experience based on the mass of the needles and a consumption of it. So we weigh it before and after. This is cool. Actually, that's a video. Let me see if I can get that to work. What's it work? So that looks like as it moves across. It's sped up. That's why it looks kind of funny. That looks like a good time. Like I'm a bit of a pyromaniac. Like I love fireworks and stuff. Like this is like a dream come true. My work sometimes is setting things on fire. I can't believe I'm getting away with this. Yeah, and publishing it too. So this is the kind of data we get. I mean, here's a basic question you can ask: how much fire energy does it take to kill a sapling? Right? You might say, well, saplings, come on, let's do real trees. I'd love to. But saplings are important. Prescribed fire, to manage fire, you might want to know, okay, I'm going to conduct a prescribed fire to reduce fire risk, but what am I going to do to this crop of saplings that I have there? I'm going to do a little ecological restoration. You might want to know, like, how much could they tolerate? So we think this is useful. And we're seeing interesting things like tolerances that don't quite line up with our expectations from mature trees, because the mature trees, you know, they have different bark thicknesses that may come into play that you don't always see in the saplings. So here we're comparing the percent mortality as we increase fire radiative energy by increasing the mass of needles we set out. And we get these sort of S-curve shaped responses, right? You can calculate LD50s from this, we do. And this is, we're comparing them: ponderosa pine, lodgepole pine, Western white pine, Douglas fir, and Western larch. And we see some surprising things like, no, this isn't surprising that Douglas fir would be so vulnerable, but we thought ponderosa pine is known as among these, a very fire tolerant species, would be sort of to the right. So this lodgepole pine, which lodgepole has this whole strategy of just, I'm going to burn up. I call it the nihilist of the forest. Burn the whole thing down and start over with serranous cones, right? But actually the saplings in our experiments were quite a little bit more resistant than we expected. Kind of cool. How about physiology? What more can we do? Can we measure what's going on inside of trees in these experiments? Certainly, and we did do that. What are the mechanisms proposed for fire-induced tree mortality? It's kind of cool. It's kind of the same story. It's called, you know, it doesn't say that explicitly on here, but we're talking about non-structural carbohydrate pools, hydraulic conductivity. We're talking about carbon starvation versus hydraulic failure all over again. It's like a whole new thing to test the same hypothesis in, right? They're filtering it here through tree growth. I don't know if I would do that, but maybe, right? Maybe, whether that leads to tree mortality. And if you follow this very niche set of literature, and I doubt that you do, not too many people are well read on this, this is kind of where what's dominated the literature for at least the last 10 years. Moving beyond the cambium necrosis hypothesis of post-fire tree mortality, cavitation and deformation of xylem in forest fires. So cool experiments showing that with enough heat, you can deform the xylem, and that can cause embolism in a bad way, right? That could be really cool. What the reviews often mix, and this is one of those fields where like people are like, you know the game. You want to get a new phytologist? They love reviews. Just keep writing those reviews. There's more reviews on this than there are experiments testing it. And they picked up on that. And then one review will say that, oh yeah, this xylem deformation was over this way, and another review picks on something wrong, and it's just been snowballing recently. But this original experiment was done with a water bath. You take an excise stem, you place it in a water bath. I'm not going to go into all the details, but you can actually, like, that's not the same as setting it on fire. And you can create a lot more of a heat impact, and you can't even test the non-structural carbohydrate effects because you've taken a piece of, I've never seen an entire tree go into a water bath for these experiments. And so reviews have kind of said, yeah, it's hydraulic failure. And I think there's a lot of influence too coming out of the drought-induced tree mortality literature. Like the two are kind of cross-pollinating each other, like in a pollution, that sort of way. You know, like it must be hydraulic failure. Yeah, that's it, right? But like the trees have plenty of water, you know, in some of these experiments. And they did in this experiment. Let me show you what we found. We have a paper that's in review right now saying, if you want to test the mechanisms of fire-induced mortality, you should use actual fire and see if that's different than a water bath. Watch this. Here's with actual fire. This was research led by Raquel Partelli-Feltrin for her PhD at University of Idaho. We measured some of the data of the non-structural carbohydrates I'll show you in a minute in my lab back when I was in Oklahoma. Published it when I got up there, so that's kind of cool. I had like ready-made collaborators up there. It was a great move. Here's the start of the experiment. This is burn day, right? Just before we burn, take some samples. With 2 experiments shown, we went back into the second one so we could have this sort of pre-fire data to show you. No difference here. And what do we see? We've got circles for the unburned, triangles for the burned. This experiment 1 has no significant differences. Oh, there's a significant difference here. What's going on? Wait, the circle is unburned. That's a higher PLC in the plant that never had any fire. 24%, it's not that high. Probably not what we think is going to come anywhere near the lethal range, which might be 60, might be 80%, depending on who you ask. If you ask me, it's 80%. So no, there's no sign of any hydraulic failure happening in these well-watered ponderosa pine saplings when we burn them at a lethal fire temperature for this species. What happened to the non-structural carbohydrates? And this is complex because I'm showing you in the needles, the stem, and the root. Before I was pooling things. Now I'm showing you separately because it's important to the story. We have in blue, unburned, in orange, burned, and then in pink, defoliated. Now, you might be noticing that there's no needle data for the defoliated treatment. Get it? Okay, we took those away, so we couldn't measure them later, right? But that's one good way to shut down photosynthesis, right? So we don't see any effect in the needles of the burned and unburned. Well, what about in the stem? Whoa, look at this by day 7. Much lower, right? In the roots, lower and consistently lower all the way till 29 days post-burn. Reduced non-structural carbohydrates. What about defoliated? Nope. Not significantly lower, at least than this one, right? It's in the middle here. So it's not about reducing the carbohydrate supply from photosynthesis. There's something going on in the roots here. They're not able to transport, right? So we think it's phloem failure, damage to the phloem that then leads to localized carbon starvation, if you will, or localized reduction of non-structural carbohydrates in the roots. We think that may play a factor in how trees die from fire. Well, this is way too simple for the kind of science I like to do. Let's get complicated. My master's student and I were like planning her thesis out, or we were like had a conversation at a coffee house or something. We're driving home, and then she says something, and I said, wait, did you just say like drought before and after fire? So we decided to do this cool experiment. You know, just kind of came out of like me not hearing what she said. That was really fun. So we decided to do this drought experiment that took 180 lodgepole pine saplings. And originally we assigned them, initially they had 3 different groups they could be in: 40 days with no water, 20 days drought, or water the whole time, 0 days drought. And then we rearranged these so that they would be equally contributing to these 2 groups. We'll either burn them, 0.4 megajoules, which should cause significant mortality in lodgepole pine, or no fire. Actually, I want to rephrase that. That doesn't cause, in our past experiments, that hasn't caused any mortality in a well-watered tree. In our previous experiment, that's why I picked it. It was kind of like on the threshold, getting close to like 50% life or death. So I picked that. And then no fire for our other treatment. And then we rearranged them again, such that they have another drought after fire, right? 40 days, 20 days, no water, or water the whole time. And so 180 saplings, 3 times 2 times 3. I know it's late in the afternoon, but not for me, guys. I'm like fired up. I'm like, finally woke up today. That's 18 treatments. We're crazy, right? 5 of the 10 in each of the 18 treatments we follow to see whether they live or die. And the other 5, we harvested them for non-structural carbohydrates and water potential measurements and some other stuff I don't quite have for you today. Here's what we saw in terms of how they died. This is a little bit confusing. Red is the burn treatments. Black is drought, no burn. If you go here, this is like water control, right? And the red meaning burned, water control again. Here's 20 days of drought before or not before burn, right? None died, no black bars. 40 days. And you can see mortality increases, right? The drought after or before fire. Mortality also seems to increase with combinations of drought before and after fire here as well. And then here as well, we have some kind of odd things like, well, we didn't get one here. Why did we have 1 die in the control? Well, there's only 5, so 20% is 1 out of our 5. So not outside the realm of fire that's, you know, half, 50% lethal, or maybe a little lower than that. So that's kind of cool. So we got significant mortality. This is enough drought. 80 days and 60 days here is enough to kill significant amounts with just drought alone. That's what the black bars mean without fire. Cool. So what was happening to water potential? Here's what I'm showing you here. We have a lot of variability going on. I'm drawn to this difference right here. I'm gonna get Drew to help me with this paper. We can get a nice significant difference here if we use just the right technique, right? Just keep trying. No, I'm not going to do that. The way we analyzed it, we do not see a significant difference here. We did here, but clearly drought is having a big effect on hydraulic failure, including in combination with fire, right? We have some trees, we saw a little something suggesting hydraulic failure here. Yeah, they had 40 days drought before we burned them. So we're showing some signs of some hydraulic failure now occurring when we don't just use well-watered trees. Use well-watered trees? I don't see that, consistent with our older experiments. How about non-structural carbohydrates? And I'll show you the roots again. And I'm kind of start with these first three ones. Look at that. Down in the roots, if you burn them, in these three treatments, right? Control, burn, control, right? Or 20 days drought, then watered, 40 days drought, then fire, or not, then watered, right? Fire brings root NSC down, all three of those, setting up just like Hakel's story in the last graph. What about these other treatments? Whoa, something interesting going on, right? We get altogether higher NSC. We don't see huge effects of the fire, maybe a little bit here, a little bit here, but not elsewhere, right? Kind of cool. So by adding the drought and the fire together, we are really confusing the situation, right? It seems a hydraulic failure in some cases is contributing more to the process than the influence of non-structural carbohydrates. With less drought or drought only before fire, we see that effect going. So I don't have a whole conclusion on what this means, but you guys are getting tired, and this is almost over. But Morgan wanted to see one more graph, right? She wants to see the sugar by starch breakdown, so I'm going to put it up here because it's kind of cool. Like this is driven by massive peak in sugar, right? Osmolar regulation in the roots. You give them drought. You want to hold on to their water, so they're able to do that somehow, perhaps by bringing starch down. But look at that y-axis; they're not scaled the same. So there may be some. Now I'm questioning. Whoa, is there still some transport of sugars from other tissues down to the roots that could drive this big boost in non-structural carbohydrates? And the sugars only go up when we give them drought after fire. Before fire, we don't get that kind of. Cool, right? There we go. So kind of wrap things all up. Trees die with drought by hydraulic failure, right? Sometimes with reduced non-structural carbohydrates before mortality. You know, people can no longer say, if I've made one thing of progress, they can no longer say this doesn't happen. We've conclusively demonstrated that it does. It's more common in some species than others, particularly in the Pinaceae conifers. But that's not what I would build my model on. I would go with hydraulic failure while we look more into, you know, if it does go down, does it matter? Hydraulic failure, we don't know. Those processes may be linked. In our field experiment, dying tree defense production was likely too late and likely constrained by low turgor pressure. So we're literally proposing, and it's going to need further testing, that the inability to build up defenses to stop bark beetle and associated fungal pathogen attack is driven more by the inability to not having water to deliver those defense chemicals to the sites of attack, not that they were constrained in their ability to store resources and carbohydrates to make these things. So we didn't see any evidence of that. I think this could be a direct one. Turns out maybe it's kind of hydraulic failure over again, right? It's a water limiting ability to defend yourself. Trees may die from fire by carbon starvation, right, in the absence of drought. But with fire plus drought, hydraulic failure starts to appear pretty important. It starts to dominate the story. More work to be done to kind of partition all of that out. Maybe this will be good enough to get that NSF grant fire. Cool. I want to acknowledge many funding sources that have supported my work over the years. As a graduate student and postdoc, and then as faculty, of course, I went to my graduate students. And acknowledge many people. I don't have all the names up here. People have helped me over the years and all this work, but Many of them are here. I think we've got time for questions. When is a human dead? How much money have we thrown at that? Right? You hear about people waking up in body bags. I mean, if we haven't spent enough money in the National Institutes of Health to give a good definition of life and death in humans, and I say haven't because I don't know if we're going to do that anymore. Oh, sorry, go there. But how could we know that in plants? I mean, it depends.
I guess so, maybe a nicer way to ask the question is, you said you took measurements right before death, so what were you using as your metric?
Sure, in the synthesis, well, when you do a synthesis and you get everyone else's data, you assume that their data is good, or that you've got enough good data that the bad data kind of washes out. And so we use their definition of death. I have a whole table in that paper that gives all the different definitions of death. And yeah, I think a better way to do a definition of death is to do something like this. Actually, don't just have like trees that survive, fully watered, those that die. Let's stop it halfway through the process and try and bring back. And when we do that, we answer your question, we get numbers. This is PLC area watering and a rewatering experiment. We get numbers around, we have a lethal dose of PLC at 80.2%. I think that's dead, but is the tree dead? The foliage is still green. So if you walk up and say, is this leaf dead? No, it's not dead. Like plants have this cool, like compartmentalized, meristem driven body plan that, you could kill off half of some species, they'll sprout right back. So like asking for the universal definition of death in trees is, anyways, that's why I gave you a hard time. It's like not the first time I heard the question. Yeah. So if they don't resprout, this might do it. Yeah.
I've got a question about the beetles. Do you think they could be sensing water potential instead of avoiding the smells and other hazards?
It could just be like a chemical arms race. Well, that we didn't see a difference, but if maybe we didn't measure the right thing.
Or they just know like it's at -3 megapascals.
Yeah, the
Go for it.
paper on. I had to miss Shaylin's defense because I was teaching or something, or I was traveling. But the paper on volatile emissions from that experiment is coming out. So yes, maybe they can smell low water potential. Yeah. And they know, I mean, whatever it is, they're good. I think that they've won the arms race. I think the trees are losing. Yeah. Clearly, if you go to these places, they look like they are, right? I mean, I've explained like monoterpenes as like, imagine, these are chemicals that are intended to defend against those bark beetles, and I imagine they evolutionarily once did, and you're smelling like toxic waste that can harm you, and you go, whoosh, like that's where that's guarding my meal, right? That's probably what's happening. I bet there's kind of a love-hate relationship. If I was a bark beetle, that's how I would feel about these things. But yeah, they may just be smelling something that's being emitted. I was showing you concentrations in the tissue.
Yeah. So
yeah, I'll get that data when I see that manuscript. Get that into the story here. Yeah. But I think we did have some differences there. I'd have to let Shaylin knows the answer. Yeah. Cool. Good question. Thank you. Yeah. Maybe. I mean, the prevailing paradigm too is that it should be about carbon. Like they can't fix the carbon, they can't build the defenses. This is like growth differentiation balance hypothesis type stuff. We don't think that's a thing in our system. Yeah. How's you?
So with the fire, it's flooring damage.
Okay.
So you think where the flooring damage happens would matter for the roots?
Yeah, I guess. Oh, like if it's high in the canopy versus low. Well, in order for us to get flowing damage high, we get even more down low. Right? So when we burn along the ground, but like we could do things. People have done stuff with like blow torches and stuff up high. Yeah, yeah. We've been thinking about like experiments where we would just lower the plant up and down and rerun it, and see if we can get different numbers above and below. And adding in girdling treatments as well to see if we can cost the same thing. Yeah, that might do it. Does that answer your question?
Yeah.
Has anybody looked at the effect on mycorrhizae or microbiome before and after fire on trees?
Maybe a little bit, but they're University of Idaho, another team led by Tara Hudeberg, just got a massive EPSCoR grant to do that. You're not an EPSCoR state here, right? You guys are ruining that for the rest of Indiana, aren't you here? Yeah, so like I'm talking multi-million, tens of millions plus to do exactly that and test it out in the field and look at what's happening. They think that that may be a very strong influence in the wild. Yeah, it's a good hypothesis. Sure. Good point.
At the beginning of your talk, you were showing that area in New Mexico that had the severe drought back in 22 years ago now. Has that abated or has it more or less continued since then?
Well, it doesn't look like this. They all fell down. And but you can go back to other sites. No, I need to get a picture. This is actually on a Native American reservation, a pueblo, if you will, in New Mexico, Native American reservation. They get permission. Craig Allen had permission to go there, I believe. So I wasn't able to go here and take this, but when I was there in 2012, for example, or even 2014 and look at it. But in this area, it doesn't look, you see, there were some wetter years that happened in the late 20 teens, and you see some green up. You get that in the inner canopy. This was just so dry that everything's dead here. The grass doesn't force. Yeah, some of the ponderosa are dead from here. And this site, I don't remember seeing after 2011 was another big drought, caused a less conscious fire in this region. Juniper mortality, but just over on another mesa, there's significant amounts of juniper mortality as a result. And you can find that in various places in New Mexico, Colorado, Utah as well. So yeah, it has continued. Yeah, at these sites.
But if it gets more moisture, do you have new young trees?
Right, that's a good question.
Repopulating it or?
No, it takes a long time to grow these things. Like those ones, where's my experiment picture? Right there. These are 20 years old. From a place with really deep soils. They had tap roots.
Yeah.
You can read anywhere you go and look about the traits of Pinus edulis. They'll tell you it doesn't have a tap root. I wouldn't have a tap root either if there was half a meter down, like hard bedrock, right? But this place had like very deep soils. They had tap roots that are growing very fast, and that's a 20-year-old pinyon pine. It's a little bit taller than me in the pot there. So I think that we've seen, and I've got other, when we look at the temperature sensitivity of the drought-induced stock and we project forward, we see trees coming off the landscape in this ecosystem faster than we can grow them back. Yeah, that's a good question for many other places where in the interim you have better growing conditions. Yeah, this is pretty slow growing. This is the edge of where you find woody plants. You get down to here, you start getting like the Sonoran vegetation, upper Sonoran, and you pick up cactus. Question. Any other questions? Yeah.
So back to the beetles. I mean, how much of the effect is the beetles versus the fungus? And are the defensive compounds kind of targeted at one or the other?
Well, that's a good question. I think that the beetles tend to come with the fungus. So we think it's acceptable to use the fungus to stimulate bark beetle attack. Right? But it isn't the same thing. We use Ophiostoma gathered actually from the Sevietta site. Turned out to be a new species. I guess every time I was talking to Robert, is that right? Simmons? Yeah, about this. He's like, oh yeah, you just go measure it and you'll find a new species. It's nothing to do. And so we think that's representative, but like you're asking, like, there's some people who say maybe, and there's this proposed, like, as a hypothesis, the fungus is growing its hyphae into the xylem, right? Gathering up spaced out nutrients where the bark beetle larvae don't go, and they're kind of farming it and eating that to get some nutrition. But with some fungi, yes, you can cause hydraulic failure. Most of the blue stain, most people I talk to don't think that causes that. I've personally gone and inoculated trees and they've been fine, but I didn't put lots in. Let me think about, there is one experiment where someone did like hundreds of inoculations to simulate bark beetle attack with just the fungus. And the mortality rate's much lower. Of course, they have drought happening at the same time. You go back and look at that. So it really does depend on the system, but most of this blue stain that's attacking Pinaceae species, they're not thought to be that fungus is actually causing death. But you can have other fungal pathogens being introduced. And I won't say that I'm not an expert on this for all bark beetles. There's so many different species of bark beetles, and they carry so many species of fungi. But There's a few people kind of pushing the hypothesis, like in spruce bark beetle I've seen, but they're not mycologists either. They're just like overly confident in their opinions. I won't say who. I'll be nice. Well, I swear that's what's happening. Using and they're showing this data with sap flow measurements. Yeah, you can also have a lack of sap flow due to drought too. Yeah. But maybe it was well watered at the time. So they're showing the fungus get in there. It's whether you could have brought the tree back at that point or not. What the bark beetle larvae do is they get in there and like girdle, they also girdle the tree. So they can cause that a carbon problem as well. We're just seeing that in this system, like the story with its confuses, it's not like other bark beetles like dendrochtonous bark beetles, at least in North America, like mountain pine beetle, western pine beetle, or even southern pine beetle, where it kills otherwise healthy trees. Those don't either until they get to really epidemic levels. I don't know if we've seen that level of outbreak in Ips confusus. It's considered more of a secondary mortality. It just sort of, some people think it shows up and takes advantage of the dying trees to reproduce. But it's happening and it's in the landscape at the same time that these other bark beetles like mountain pine beetle, once that thing gets rolling at full epidemic, it chooses the healthy trees to kill. Like it's, talk about smelling good trees. It's like, yeah, this is a good place to raise my young until it wipes out everything above at certain levels above 10 centimeters of diameter. Yeah. Yeah, good question. So yeah, it's going to be very system dependent, the curse of context dependency in ecology. Yeah. One more? Okay, if anybody needs to split, that's okay. I won't feel rude.
Okay, so you mentioned how like induced response decreases with drought, right? But the defense molecules are the same even when they're in drought.
Yeah.
I'm assuming the induced response is producing those defense chemicals.
I should have made that more clear. Yeah, good question. You have both constitutive and induced responses to defense, right? So these trees are going in. Like when I show you a time series, like at the end, these were all constitutive data. Well, that's way at the end. The stuff I didn't show. About like we'll do these needle monitor because this is all constitutive. Thanks for asking the question, not induced. So we do have some fungal responses. They don't we do have induced responses to fungal inoculation in some of these trees. They don't seem to explain who would then later die or live either, although we only did that on a subset.
Okay, so it's not that the ones in drought aren't giving like more constitutive defense, like they're not building up their defenses. So I don't.
Right, they're not. Yeah, they're not building up constitutive defenses, but they weren't really having much of an induced response at this field site. Anyways, in the experiment, we saw the induced response in the less water stress trees than we did experiencing the water stress in the greenhouse experiment, not the field experiment. Yeah, you know, I do lots of seedling and sapling and growth chamber type experiments. I've done that too, and everyone's, oh, you know, you try to publish that. Well, the reviewer says, what's this? You can't learn anything because it's not in the real world. Then you go out and try to do it in the real world, and it's hard. You get lucky like this, you get inconclusive data because, you know, look at these trends, right? Things are this is the challenge. When you think about other than non-structural carbohydrates, I mean, what, you know, things are moving around. They don't stay still. Trees are dynamic, right? And so you really do sacrifice a lot of control in these kinds of experiments. I think the best way is to kind of bring from both. That's a good question. Is the constitutive. Cool. If anyone's got another question, I'll hang out for a bit.
The legacy. Under